Welcome back to the deep dive where we unpack complex topics and try to extract those golden nuggets of knowledge.
Ready to dive in?
Today, we're exploring how genes provide the instructions for building proteins using excerpts from a college-level genetics lecture as our guide.
It's a fascinating journey from DNA to those workhorse molecules that carry out such a huge variety of functions in our bodies.
It's like DNA is a recipe book, and we're learning how to read it. You might recall Beadle and Tatum's Nobel Prize-winning work that solidified the link between genes and proteins.
Absolutely. Their experiments with that bread mold showed that a mutation in a gene could lead to a specific enzyme deficiency.
It's mind-blowing how they used bread mold to figure that out.
Yeah.
So essentially, they proved that genes hold the instructions for building proteins, which act like tiny molecular machines, keeping everything running smoothly.
Precisely. They drive everything from digesting your food to fighting off infections. And it was scientists like Archibald Garrod in the early 20th century who started to piece together this connection between genes and enzymes.
It's like figuring out that a missing ingredient in a recipe leads to a completely different dish. Garrod's work on inherited metabolic disorders was a huge leap forward.
Indeed. And to understand how this intricate process works, we need to delve into the flow of genetic information, often called the central dogma.
Okay, let's unpack this dogma. I remember learning about transcription and translation, but it would be great to have a refresher.
Of course, transcription is the process where DNA is copied into messenger RNA or mRNA. Think of it as creating a temporary working copy of the genetic instructions.
And then translation comes into play.
Exactly. Translation is where that mRNA molecule is used as a template to assemble amino acids, the building blocks of proteins, into a specific sequence.
So we have this elegant flow of information, DNA to RNA to protein. And all of this is orchestrated by molecular machines like RNA polymerase and ribosomes, right?
Right. RNA polymerase is like a diligent worker, carefully copying the DNA instructions onto an mRNA molecule during transcription.
I can almost hear those tiny molecular gears turning. But how does RNA polymerase know where to start and stop copying?
There are specific DNA sequences that act like signals. For instance, RNA polymerase binds to a promoter region on the DNA, marking the starting point for transcription.
It's like finding the first line of the recipe in our DNA cookbook.
And just like a recipe has an end, there are terminator sequences on the DNA that signal RNA polymerase to stop transcribing.
So the mRNA molecule essentially carries a transcribed version of the gene ready for translation.
Right. It's like a messenger carrying vital instructions from the nucleus to the ribosomes in the cytoplasm where protein synthesis takes place.
It's fascinating how this whole process is compartmentalized in eukaryotic cells. But before we move on to translation, I'm curious, are there any key differences in transcription between prokaryotes and eukaryotes?
You've hit on an important point. In prokaryotes, which lack a nucleus, transcription and translation happen simultaneously. It's incredibly efficient.
Wow, so it's all happening in the same compartment.
Exactly. But in eukaryotes, transcription takes place in the nucleus, and the mRNA transcript undergoes some processing before it's shipped out to the cytoplasm for translation.
Makes sense. It's like having separate departments for recipe copying and protein assembly.
And that processing is crucial. For instance, in eukaryotes, the mRNA molecule gets a 5-foot cap and a poly-A tail. These modifications protect the mRNA from degradation and help it bind to ribosomes.
Like adding a protective cover to our recipe copy before sending it out into the busy cytoplasm.
Precisely. And there's another crucial step called RNA splicing. This is where non-coding sequences called introns are removed from the mRNA, leaving only the protein coding sequences, or exons.
It's like editing out the unnecessary parts of the recipe before we start cooking.
Right. And what's fascinating is that this splicing process can lead to a single gene producing multiple different protein variants depending on which exons are included.
It's like having multiple recipes from a single set of instructions.
Yeah.
That adds a whole new layer of complexity. So we've got this processed mRNA molecule ready to go. Now, how does the sequence of nucleotides in DNA actually specify the sequence of amino acids in a protein?
That's where the genetic code comes in. Scientists have cracked this code and discovered that it's based on triplets of nucleotides called codons.
Codons, those are groups of 3 nucleotides, right? Like a 3-letter word in the DNA language.
Exactly. Each codon specifies a particular amino acid or acts as a signal. For example, the codon AUG not only codes for the amino acid methionine, but also serves as the start signal for translation.
So AUG is like the once upon a time at the beginning of the protein story.
I like that analogy. And there are also 3 stop codons that signal the end of the protein coding sequence. Think of them as the end of our protein story.
So with 4 different nucleotides and 3 nucleotides per codon, that means there are 64 possible codons. But I remember there are only 20 different amino acids.
You're right. The genetic code is redundant, meaning that multiple codons can specify the same amino acid. For example, the amino acid leucine is coded for by 6 different codons.
So it's like having synonyms in the DNA language.
Yeah.
Does this redundancy serve any purpose?
It does. This redundancy helps to minimize the impact of some mutations. If a mutation changes a codon to one that specifies the same amino acid, there's no effect on the protein sequence.
That's a clever safety mechanism. So it's like having a typo in the recipe, but it doesn't actually change the final dish.
Precisely. But as we discussed earlier, not all mutations are harmless. Some can change the amino acid sequence of a protein, altering its structure and function. This can lead to a range of consequences, from minor variations to serious diseases.
Right, like sickle cell anemia, which is caused by a single nucleotide change that affects the shape of red blood cells. It's a powerful example of how even a small change in the DNA sequence can have a significant impact.
It really highlights the importance of understanding the genetic code and the potential consequences of mutations.
It's amazing how such tiny changes at the molecular level can have such profound effects on our health and well-being.
Indeed, and it underscores the elegance and complexity of the molecular world.
Speaking of complexity, I'm wondering, are there any exceptions to this central dogma of DNA to RNA to protein?
That's a great question. While the central dogma holds true for most organisms, there are fascinating exceptions, particularly in the world of viruses.
Viruses, those tiny infectious agents that can make us sick, don't some of them have RNA as their genetic material instead of DNA?
You're exactly right. Some RNA viruses use an enzyme called reverse transcriptase to synthesize DNA from an RNA template. This challenges the traditional flow of genetic information.
Reverse transcriptase. It's like a molecular rebel breaking the rules of the central dogma.
Indeed, HIV, the virus that causes AIDS, is a classic example of a retrovirus that uses reverse transcriptase.
So even in the world of molecular biology, there are always exceptions to the rules.
Absolutely. And the study of viruses has revealed many surprises and challenges to our understanding of genetics.
This deep dive is already making me rethink everything I thought I knew about genes and proteins.
Wow.
But let's shift gears a bit and focus on translation in more detail. We mentioned ribosomes earlier, but what are these tiny molecular machines actually made of?
Ribosomes are remarkable structures composed of both RNA molecules and proteins. They have 2 subunits, a large one and a small one, that come together during translation.
So it's a combination of RNA and protein working together to build more protein. That's pretty meta.
Isn't it? And the structure of ribosomes is highly conserved across all life forms, emphasizing their fundamental importance in protein synthesis.
I'm picturing these ribosomes as tiny construction crews bustling around the cell assembling proteins based on the mRNA blueprints.
That's a great analogy. And just like any construction site, there are specific tools and processes involved in building a polypeptide chain. Let's break it down step by step.
Right.
First, the mRNA molecule carrying the genetic code binds to the small ribosomal subunit.
Okay, so that's the first step.
Then a special initiator tRNA carrying the amino acid methionine binds to the start codon on the mRNA.
So that's the beginning of the polypeptide chain.
Exactly. Next, the large ribosomal subunit joins the complex, forming a functional ribosome ready for action. Think of it as the construction crew assembling and getting ready to work.
Got it. So the ribosome is now fully formed and ready to translate the mRNA code into a protein. What happens next?
Now, the ribosome has 3 important binding sites for tRNA molecules: the A site, the P site, and the E site.
Okay, those sound a bit technical. Can you explain what each of those sites does?
Of course. The A site is where the incoming tRNA carrying the next amino acid to be added to the polypeptide chain binds to the mRNA. The P site is where the tRNA holding the growing polypeptide chain is located, and the E site is where the tRNA that has delivered its amino acid exits the ribosome.
So it's like a 3-step process. Incoming tRNA in the A site growing chain in the P site and exiting tRNA in the E site.
You got it. Now, during elongation, a new tRNA molecule carrying the next amino acid in the sequence enters the A site. The ribosome then catalyzes the formation of a peptide bond between the amino acid in the A site and the growing polypeptide chain in the P site.
So we're linking amino acids together one by one, following the mRNA instructions.
Precisely. And then something fascinating happens. The ribosome shifts down the mRNA molecule by 1 codon. This movement, called translocation, repositions the tRNAs. The tRNA holding the growing polypeptide chain moves from the P site to the E site, and the tRNA in the A site moves to the P site, leaving the A site open for the next tRNA.
That's like a molecular dance with the tRNA molecules moving between the different binding sites on the ribosome.
Exactly. And this process of elongation continues codon by codon until a stop codon on the mRNA reaches the A site.
The end of our protein story. What happens then?
When a stop codon enters the A site, a protein called a release factor binds to it instead of a tRNA. This signals the ribosome to release the completed polypeptide chain.
Mission accomplished. Our protein is assembled.
Well, almost. The polypeptide chain still needs to fold into its proper three-dimensional shape to become a functional protein.
Right, like protein origami, where the chain twists and turns into a specific shape. Didn't we discuss that this folding is crucial for the protein to function correctly?
You're exactly right. Without the correct shape, the protein won't be able to perform its job.
This whole process of translation is so intricate. It's amazing to think that all of this is happening constantly in every cell of our bodies, millions of times per second.
It's a testament to the elegance and complexity of the molecular world. And we're only just beginning to scratch the surface of understanding all of its intricacies.
Speaking of intricacies, what happens if something goes wrong during translation? What if there's a problem with the ribosome or the tRNA molecules?
That's a great question. Errors during translation can lead to the production of misfolded or truncated proteins, which can have a variety of consequences for the cell.
Like what kinds of consequences?
Well, imagine misfolded proteins as causing a traffic jam inside the cell. They can accumulate, leading to cellular stress and even cell death. They can also trigger an immune response as the body recognizes them as abnormal.
That sounds like a serious problem.
It can be. There are also genetic disorders caused by mutations in genes that code for ribosomal proteins or tRNA molecules.
So a defect in the machinery itself can disrupt the process.
Precisely. These mutations can disrupt the process of translation, leading to developmental problems and other health issues.
Wow. It's clear that proper translation is essential for cellular function and overall health.
Absolutely. And it's just one piece of the complex puzzle of gene expression. There's a whole other layer of control that ensures the right genes are expressed at the right time and in the right cells, called gene regulation.
We touched on this earlier with the trap and lack operons in bacteria. But how does gene regulation work in eukaryotic cells, which are much more complex?
Eukaryotic gene regulation is a fascinating and intricate process with multiple layers of control. Remember, in eukaryotes, DNA is packaged into chromatin, which is a complex of DNA and histone proteins.
Right, I remember learning about that. It's like the DNA is wrapped around these histone proteins, forming these bead-like structures.
Exactly. And the structure of chromatin can influence gene expression. Chromatin can be either tightly packed, making it difficult for transcription factors to access the DNA, or more loosely packed, allowing for easier access.
So it's like having a library with bookshelves that can be moved closer together or farther apart, making it harder or easier to find the book you're looking for.
A perfect analogy. And this chromatin structure can be modified by enzymes that add or remove chemical groups to the histones, a process called histone modification. These modifications can act like switches that turn genes on or off.
Wow, so it's like having a way to fine-tune gene expression without changing the DNA sequence itself.
Precisely. And there's another layer of regulation called DNA methylation, where a methyl group is added to a cytosine base in the DNA sequence. This often leads to gene silencing, preventing transcription.
So it's like adding a do not disturb sign to a gene. Fascinating.
It is. And these epigenetic modifications, which include both histone modifications and DNA methylation, can be influenced by environmental factors such as diet stress and exposure to toxins.
Wait, so that means our environment can actually influence how our genes are expressed.
Yeah.
It's incredible.
It is. It's a fascinating area of research that's shedding light on the interplay between nature and nurture. But let's move beyond epigenetics and talk about the role of transcription factors in eukaryotic gene regulation.
Okay, what are transcription factors?
They're proteins that bind to specific DNA sequences and either activate or repress transcription. Think of them as the conductors of the cellular orchestra directing which genes are expressed.
That makes sense.
Yeah.
Different genes need to be expressed in different cells and at different times.
Exactly. And the combination of transcription factors present in a cell determines which genes are turned on or off, leading to the unique gene expression profile of each cell type.
This is all starting to make sense. So we've got chromatin structure, epigenetic modifications, and transcription factors all playing a role in regulating gene expression in eukaryotes.
Precisely. And we haven't even touched on post-transcriptional regulation, which involves controlling the processing stability and translation of mRNA molecules.
Wow, there's a whole other level of regulation happening after transcription. Tell me more.
For example, alternative splicing, which we mentioned earlier, allows a single gene to produce multiple different protein variants.
Right, that's where different combinations of exons are included in the final mRNA.
Exactly. And there's another fascinating process called RNA interference, or RNAi, where small RNA molecules can bind to mRNA molecules and either block their translation or trigger their degradation.
It's like having a team of editors that can fine-tune the mRNA transcripts before they're translated into proteins.
Precisely. So as you can see, eukaryotic gene regulation is a complex and dynamic process involving multiple layers of control that ensure the right genes are expressed at the right time and in the right cells.
It's truly remarkable. But I'm curious, why is all of this regulation necessary? What's the evolutionary advantage of having such a complex system?
That's a great question. One of the key advantages is flexibility. By having multiple levels of control cells can fine-tune gene expression in response to changing environmental conditions.
So it's like having a sophisticated control panel that allows cells to adjust their gene expression based on the situation.
Exactly. Another advantage is efficiency. By only expressing the genes that are needed at a particular time, cells can conserve energy and resources.
That makes sense. It's like turning off the lights when you leave a room so you don't waste electricity.
Precisely. And this efficient use of resources is crucial for the survival and reproduction of organisms. So as you can see, eukaryotic gene regulation is a marvel of evolution, providing both flexibility and efficiency in controlling gene expression.
It's truly awe-inspiring. We've covered a lot of ground today exploring the intricate journey from genes to proteins. We've delved into the molecular mechanisms of transcription, translation, and gene regulation, and we've even touched on the fascinating world of mutations and epigenetic modifications.
It's been a whirlwind tour of the cellular world.
It has,
All right.
and I hope our listeners are starting to grasp just how much is going on inside their cells every second of every day. So as we wrap up part one of this deep dive into the fascinating world of genes and proteins, I want to leave you with one final thought.
Yes.
We've seen how our genes provide the blueprint for who we are, influencing our traits, our health, and even our behavior. But we've also seen how gene expression is not fixed. It can be influenced by our environment, our experiences, and even our choices.
You're absolutely right. Our genes are not our destiny.
So as you go about your day, remember that you are not simply a product of your genes. You have the power to shape your own destiny at the molecular level and beyond.
That's a powerful message to end on. Join us for part 2 of this deep dive, where we'll unpack even more about the amazing world of genes and proteins. Welcome back to our deep dive into the fascinating world of genes and proteins. I'm eager to continue our exploration.
Me too. Last time we laid the groundwork exploring how DNA provides the instructions for building proteins.
We journeyed from the nucleus, where DNA is transcribed into messenger RNA, to the ribosomes and the cytoplasm, where that mRNA is translated into a chain of amino acids.
Right, and we even touched on how this intricate process can go awry, leading to misfolded proteins and potential health problems. It's amazing how much is happening at the molecular level every second of the day.
It really is.
Hmm.
But remember, even with all this complexity, the cell has elegant control mechanisms in place to regulate gene expression. We briefly discussed operons in bacteria, but let's delve deeper into how gene regulation works in eukaryotic cells.
I'm all ears. Eukaryotic cells with their membrane-bound organelles seem so much more complex than bacteria.
They are, and their gene regulation is multi-layered, involving a complex interplay of DNA sequences, regulatory proteins, and those fascinating epigenetic modifications we talked about.
Right, epigenetics is like the cell has a way to fine-tune gene expression without changing the underlying DNA sequence itself.
Exactly. Think of it like this. You have a cookbook with all the recipes, but you don't want to make every dish every day.
So epigenetic modifications are like deciding which recipes to make based on what ingredients are available or what you're in the mood for.
A great analogy. These modifications can be influenced by environmental factors like diet stress and exposure to toxins.
So our environment can actually influence how our genes are expressed.
Precisely. It highlights the dynamic interplay between our genes and our experiences.
That's mind-blowing. But let's back up a bit. You mentioned that in eukaryotes, DNA is packaged into chromatin, which is a complex of DNA and histone proteins.
Yes. Remember those bead-like structures we talked about? That's chromatin. And the structure of chromatin can actually influence which genes are accessible for transcription.
It's like having a library with bookshelves. They can be moved closer together or farther apart, making it harder or easier to find the book you're looking for.
A perfect analogy. And the accessibility of those books or genes is influenced by histone modifications.
Right, those are the chemical changes to the histone proteins that act like switches turning genes on or off.
Exactly. And there's another layer of control called DNA methylation, where a methyl group is added to a cytosine base in the DNA sequence. This often leads to gene silencing, effectively turning the gene off.
So it's like adding a do not disturb sign to a gene. Fascinating. It seems like every time we peel back a layer of this process, we find even more intricate control mechanisms.
That's the beauty of molecular biology. And we haven't even talked about transcription factors, which play a crucial role in eukaryotic gene regulation.
Okay, remind me again, what are transcription factors?
They're proteins that bind to specific DNA sequences and can either activate or repress transcription. Think of them as the conductors of the cellular orchestra directing which genes are expressed.
So they're like the decision makers deciding which genes get read and which ones stay silent?
Exactly. And different cells have different sets of transcription factors, which allows them to express different genes and carry out specialized functions.
That makes sense. A muscle cell needs to express different genes than a nerve cell.
Right, precisely. And the combination of transcription factors present in a cell determines which genes are turned on or off, leading to the unique gene expression profile of each cell type.
Wow, it's amazing how all of these different factors work together to regulate gene expression in eukaryotes. We've got chromatin structures, epigenetic modifications, and transcription factors all playing their part.
It's a beautifully orchestrated system, and we haven't even touched on post-transcriptional regulation, which takes place after the mRNA molecule has been transcribed.
Wait, there's even more control after transcription. I am all ears.
Absolutely. Remember alternative splicing, where different combinations of exons can be included in the final mRNA molecule?
Right. That's how a single gene can code for multiple protein variants.
Exactly. And there's another process called RNA interference, or RNAi, where small RNA molecules can bind to mRNA molecules and either block their translation or trigger their degradation.
It's like having a team of editors that can fine-tune the mRNA transcripts before they're translated into proteins.
Precisely. So you see, eukaryotic gene regulation is a multi-layered and dynamic process.
It's truly remarkable how much control the cell exerts over gene expression. But why is all this regulation necessary? What's the evolutionary advantage of such a complex system?
That's a great question. One of the key advantages is flexibility. By having multiple levels of control, cells can fine-tune gene expression in response to changing environmental conditions.
It's like having a sophisticated control panel that allows cells to adjust their gene expression based on the situation they find themselves in.
Exactly. And another advantage is efficiency. By only expressing the genes that are needed at a particular time, cells can conserve energy and resources.
That makes sense. It's like turning off the lights when you leave a room so you don't waste electricity.
Precisely. And this efficient use of resources is crucial for the survival and reproduction of organisms. So as you can see, eukaryotic gene regulation is a marvel of evolution, providing both flexibility and efficiency in controlling gene expression.
It's like the cell is a master conductor orchestrating a symphony of gene expression.
I like that. A symphony with many instruments all playing their part to create a harmonious whole.
It's mind-blowing to think about all of this happening inside our bodies right now. So we've explored how DNA is transcribed into mRNA, how mRNA is translated into proteins, and how this whole process is regulated in intricate ways.
We've covered a lot of ground, but remember, we're still just scratching the surface of this vast and fascinating field.
That's right. There's so much more to learn. For instance, we've talked about how the genetic code is based on triplets of nucleotides called codons. But what happens when there's a change, a mutation in that code? What are the implications for protein structure and function?
Mutations. They're the source of variation, the drivers of evolution. And they can have a profound impact on the proteins that are produced. Let's delve into the world of mutations and explore their consequences.
Lead the way. I'm ready to dive deeper. Welcome back to the final part of our deep dive into the intricate world of genes and proteins.
It's been quite a journey exploring the molecular mechanisms that, drive life as we know it.
We've unpacked how DNA is transcribed into messenger RNA, how that mRNA is translated into a chain of amino acids, and how this whole process is regulated with incredible precision.
And last time we left off with a cliffhanger talking about mutations, those changes in the DNA sequence that can have a ripple effect on protein structure and function.
Right, mutations are like typos in the genetic code. Sometimes they're harmless, but sometimes they can have, significant consequences.
Exactly. Imagine a recipe where a single letter is changed. Sometimes it might not make much of a difference, but other times it could completely alter the dish.
So some mutations are silent, having no effect on the protein.
Precisely, these silent mutations often occur in the third position of a codon. Remember how the genetic code is redundant with multiple codons specifying the same amino acid.
Right, so even if a mutation changes the DNA sequence, it might not change the amino acid that's incorporated into the protein.
Exactly, but other mutations can have more noticeable effects. For instance, a missense mutation changes a single amino acid in the protein.
And that could alter the protein's shape and function.
Precisely, think of it like swapping out a key ingredient in that recipe. It might still be edible, but the flavor and texture could be completely different.
And then there are nonsense mutations, which introduce a premature stop codon, cutting the protein short.
Right. It's like ending the recipe abruptly before all the steps are completed. The resulting protein is often truncated and non-functional.
That sounds problematic.
It can be, and we can't forget about insertions and deletions where nucleotides are added or removed from the DNA sequence.
Right, those can cause frame shift mutations, shifting the reading frame of the genetic code and leading to a completely different amino acid sequence.
It's like adding or deleting a letter in a sentence, completely changing the meaning of the words that follow.
Wow, mutations can really shake things up at the molecular level. But aren't mutations also the driving force of evolution?
Absolutely. Mutations introduce variation into the gene pool, providing the raw material for natural selection to act upon.
So some mutations might actually be beneficial, giving organisms an advantage in their environment.
Exactly. Those beneficial mutations are more likely to be passed on to future generations, leading to evolutionary change over time.
It's incredible how these tiny changes in DNA can have such profound effects on individuals and even entire species.
It is, and studying mutations helps us understand the complexities of genetic diseases, develop new therapies, and even trace the evolutionary history of life on Earth.
Speaking of genetic diseases, we've touched on how mutations can disrupt the process of protein synthesis. But what about the broader impact of mutations on human health?
Mutations are implicated in a wide range of diseases, from cancer to cystic fibrosis.
So a mutation in a gene can disrupt the normal function of a cell, leading to disease.
Precisely. For example, in cancer, mutations can activate oncogenes, which promote uncontrolled cell growth, or inactivate tumor suppressor genes, which normally keep cell division in check.
So it's like having a car with a stuck accelerator and broken brakes.
A great analogy. And in cystic fibrosis, a mutation in the CFTR gene disrupts the transport of chloride ions across cell membranes, leading to the buildup of thick mucus in the lungs and other organs.
It's amazing how much we've learned about the genetic basis of diseases. This knowledge is crucial for developing new treatments and therapies.
Absolutely. And with advances in gene editing technologies like CRISPR-Cas9, we're entering a new era of possibilities for treating genetic diseases.
CRISPR sounds revolutionary. Didn't we touch on that briefly earlier in our deep dive?
Yes, CRISPR-Cas9 is a powerful tool that allows scientists to precisely edit genes. It's like having molecular scissors that can cut and paste DNA sequences.
It's like we're entering the age of genetic surgery.
In a way, yes. CRISPR-Cas9 holds immense potential for correcting genetic defects, developing new disease therapies, and even engineering crops that are more resistant to pests and diseases.
Wow, the possibilities seem endless. But with great power comes great responsibility,
Mm.
right? There are definitely ethical considerations when it comes to gene editing.
Absolutely. It's crucial to have open and honest conversations about the potential benefits and risks of these powerful technologies.
We need to ensure that gene editing is used responsibly and ethically for the betterment of humanity and the planet.
I couldn't agree more. It's an exciting time to be in the field of genetics, but we need to proceed with caution and wisdom.
Well, I think we've reached the end of our deep dive into this fascinating world of genes and proteins. We journeyed from the nucleus to the cytoplasm, from DNA to RNA to protein, exploring those intricate mechanisms of transcription, translation, gene regulation, and even the impact of mutations.
It's been an incredible journey, and I hope our listeners have gained a deeper appreciation for the elegance and complexity of the molecular world.
I hope so too. So until next time, keep exploring, keep questioning, and keep learning. The world of science is full of wonders waiting to be discovered.